classification algorithm which is able to classify node labels, to classify instances in the case in which we have multiple tasks at the same time. We have to learn multiple tasks at the same time. Often when we have a classification problem, we can solve it independently from other classification problems. But when we have that our problems are related to each other, we may classify them at the same time in order to improve the accuracy of our prediction. The context, the setting I'm going to use is a semi-supervised setting based on a graph in which our instances are nodes in this graph, and their similarities are encoded in the connections among instances. And we have some nodes which already have a label. That's our training set. And that are those in this graph which are black and light blue. And meaning that, for example, in this case, light blue is a positive instance for the class we are going to predict, for the task we are going to predict. The black one are the negative one. The white node instead are unlabeled nodes. That are the nodes for which we want to infer a prediction, black or light blue, according to the neighborhood of these nodes in the graph. And so if we have two different tasks, as in the example, on the same graph, because the connections and the nodes are the same, we may predict each task independently from each other and infer a kind of classification like this one that we see in the graph. Starting from the initial condition, according to any kind of prediction algorithm, we can reach a situation like this one. Our aim is, in the case in which the two tasks are related to each other, how we can improve independent prediction. So this is our input. We have the graph of instances. We're supposed to have n instances, and their connection that we're supposed to be a symmetric connection. And the weight of this connection is 0 if there is no connection among two instances. And the higher, of course, the weight, the more similar are two instances in the graph. On the other side, we have a graph of tasks. We know that some of the tasks we are going to predict, they are related to each other. We encode this information in the matrix C that for simplify the problem, we're supposed to be in the 0-1 interval. 1 is a similar task, 0 is a not related task. And of course, we want to use this information while predicting each task. So what we want to do, we want to use a Bayes learner and then try to exploit our multitask information. The Bayes learner we adopt is a level propagation algorithm. Why we chose this algorithm? Because it's a well-known algorithm which performs quite well in very different tasks, also in a variety of different problems, and also it's efficient. It's an efficient algorithm. It computes, given the information from the neighborhood of an unlabeled node and the labels for his labeled neighborhood, it computes a function f that to some extent is a kind of average of the neighborhood signal. This method supposes that the labels, the known labels, are binary, -1 for example, for a negative, and 1 + 1 for positive nodes. And the aim is to compute, we encode these labels in the vector y, and the aim is to compute another vector f, which is a kind of membership function. It's a real vector such that the higher the score this vector assigned to an unlabeled node, the higher the likelihood that this node would be a positive for the task we are going to predict. And f is computed as the argument that which minimizes this objective function, in which the constraint means that for any labeled node, the score f we predict should be exactly the label we know for that node. While we don't have constraint on the unlabeled node, and minimizing these objective functions means minimizing the cut size of the graph. That is the sum of the weights connecting two nodes with different labels. In this objective function, L is the Laplacian of the graph connection of W, of the matrix we call W in this slide. Optimizing this problem, the solution has also a closed form that you can see here, that is useful when the connection matrix is not singular and you can compute the inverse. But there is also an iterative version of this algorithm which is also quite fast and has a fast convergence. And in this slide, just we denote by U the set of unlabeled nodes and by S the set of labeled nodes, and f underscore U is the subvector associated to unlabeled nodes. So that's what label propagation does. Now we want to extend this approach in order to exploit also task relationships. Usually, when we have a graph of tasks, the approach is to try to constrain the labeling associated to each task to be closer when the tasks are related to each other. It's something quite obvious. And we tried this approach in a specific problem that I will describe later, in predicting protein functions for fly and human organisms. And what we got is that this approach doesn't work in this case. Here we mention this algorithm as MTLP INV, while LP is the approach, the label propagation method that I described just before. And this was quite curious for us because we didn't expect such results. Here I don't report how we compute such kind of multitask algorithm. It will be clear from the next slides when I will talk about our method. And so, what we want to do, what we did, we tried the complementary approach. I don't know how to define it. Instead of requiring that similar tasks have similar labelings, we require that dissimilar tasks have dissimilar labelings. It seems the same, but it's not. It's not the same. Here I report the average result we got by comparing the base learner LP, our method, that is the yellow one, that we called MTLP, multitask label propagation, and the previous approach that I showed, MTLP INV. And as you can get, the red bar, that is the multitask usual approach that I described in the previous slides, is always the lowest performance. While our algorithm, the yellow bar, achieves better performance compared to both in all our experiments. It's something quite frequent in our experiment. We never get different results, different trends. And so I want to try to briefly describe how our method works and why we get this kind of results. Here I want to say, I should say that LP is not the standard label propagation algorithm that I previously described. This is already an improved algorithm to manage data imbalance, because the experiment we did, we adopted very unbalanced labelings. That is, we had a task with very few positives and much more negatives. For example, 10 positive and 10,000 negatives. And in such kind of setting, most of learning algorithms poorly performs, because they tend to learn just the negative class. And so we adopt a kind of imbalance-aware regularization for LP-based method. And these are the results of this already improved algorithm. So this is our method. First, we need a task dissimilarity matrix that, since we called in our setting C as the similarity matrix for task, we call our dissimilarity matrix as overline C, just to distinguish these two metrics. And so given for any couple of tasks a measure of dissimilarity among these two tasks, which is encoded in the matrix overline C, we compute the Laplacian of C, that we call overline A, and we apply this transformation. If Y is the label matrix, that is, each column of this matrix is the labeling for one task, we apply this transformation, that is a matrix transformation between Y, the label matrix, and our graph Laplacian overline A. And this is what we use. We use overline Y as new labelings for our label propagation. This is, after we transform labelings according to the multitask map, we just solve this problem, which is, if we don't consider here overline Y, but just Y, the original label matrix, this is just a single task label propagation repeated over all the tasks independently. Here instead we use in our constraint for label nodes our transformed labelings, which are not anymore in -1, 1 set. They are real labels now. And then we compute this kind of solution, the argument which minimizes this objective function, and surprisingly this approach works. It's so simple, but we try to understand why we have this kind of result. And the reasons are essentially two. The first one is that this kind of map increases the distance between dissimilar tasks, between the labelings associated to dissimilar tasks. If we consider the columns of the matrix Y associated to two different tasks, and we compute their norm, for example, L2 norm, and then we apply our transformation and compute again the L2 norm between these two vectors, their L2 norm increased. So the similar tasks are associated with more distant labelings in this sense. And this is why one contribution of our algorithm, that is the one which make difference between LP, the blue bar and the yellow bar in this graph. The other contribution, the other reason is number 2, is that this transformation is suitable for unbalanced problem. When you have a rare positive in your labeling, in your classification problem, this kind of transformation is suitable for such kind of labelings, for problems in which positive are rare. And so what happened that when you have such kind of labelings, usually in the propagation of labels, negative labels overwhelm the positive ones. This is what any kind of learner perform. But after our multitask transformation, this doesn't happen. It's positive labels can propagate more than the base LP method. I report here a slide in order to show this behavior, why this happened. Here we have the initial label matrix. Each column is a task. We're supposed to have 4 instances, which are in this graph, view 1, view 4, and we suppose that 2 of these instances are negative, the 4th one is positive, and we suppose that we don't know the label of the first instance and we want to predict it. And suppose that the true label for view 1 are given here, which is a negative for the first 2 tasks and positive for the third one. We suppose that all tasks are dissimilar to each other. We suppose that the graph of task is a click. Over line C is a click in this case. The corresponding Laplacian is this one. If you apply our multitask transformation, you get this new label matrix. You can see how the labelings has been expanded from each other. And why now our algorithm can predict positive labels also when the labels are rare? Suppose now we want to predict for the third task the label of the first node. LP method, the LP base method, since here V1 is connected to all the other 3 nodes, it sums the labels of all these neighbors, that is -1, -1, +1, and the result is -1. Label propagation wrongly predict the label of V1 because V1 for this task is 1, is positive. But our method, after the transformation of this one, you sum 5 minus 1 minus 1 is +3. The label is positive, is correctly predicted. So this is we formally prove these 2 properties in our paper. Here is just an example to give an idea of why this simple approach achieves better results of both LP and of the other approach in which you make closer similar tasks. The other approach you can get by simply changing this transformation, instead of considering over line A, to consider its inverse matrix. In that case, you can make closer tasks instead of more far those tasks. So the approach is almost the same, but that one doesn't work for the reason I show, because if you consider the inverse matrix here of overline A, you don't get this kind of behavior. You get instead of labels higher than 1, you get labels lower than 1. So you make worse the prediction. So that's in order to test this kind of properties, we found a problem which is characterized by very unbalanced labelings. We consider the problem of predicting protein functions in some model organisms. Here I report the Drosophila melanogaster and the Homo sapiens proteins. Among parentheses you can see the number of proteins we consider. To some extent we consider genome-wide approach, just excluding proteins who doesn't have any positive annotation for any GO term, because we have to validate our model, and so we consider just proteins which has at least one positive annotation for all GO terms. And then we consider all the gene ontology terms which has annotation between 5 and 100 positive annotations, in order to consider the most specific terms in the ontology, which are those that also better describe protein function, they are more specific. And we got thousands of GO terms. There are thousands of tasks in this setting. I want to remark that this setting is a very general setting. In our experiment, each node is a protein. The connection are any kind of protein similarity that has been computed by considering different kind of experiments, and a link here can be the integration of different information among proteins. But it can be also, for example, each node can be a drug in the drug repositioning problem, in which you have connection among drugs, and the task to be predicted is a drug category from drug bank, for example. Or it can be, each node can be a gene, and the task can be a disease for which you want to determine which are the causative genes for that disease. This problem is called gene disease prioritization. And recently many approaches has been proposed to compute the similarities among diseases, and consequently dissimilarity among diseases. So the problem is quite general, and it can find many applications in bioinformatics. We focus on protein function predictions, and we compute some similarity and dissimilarity measures for GO terms by using standard methods in literature. We adopt a Lin and Jiang measure, and also an annotation-based measure which does not take into account the ontology of the structure of gene ontology. This kind of measure, for example, simply given two different functions, consider as a similarity the number of shared genes, that is, genes annotated with both functions, divided by the total number of genes annotated for these two functions. That is a kind of standard annotation-based similarity measure. And also we compare, we test other state-of-the-art algorithms for graph-based prediction, semi-supervised graph-based prediction. Here we have the results where RW is the random walk algorithm, GBA is the guilt by association algorithm, KNN is the K nearest neighbor algorithm. COSNET is a recently proposed algorithm based on OFI networks, which has been designed for unbalanced problems, specifically designed for unbalanced problems. And it has been compared with the top methods of several challenges like mouse Funk1 competitions for gene function prediction. And for this reason I report also this method, because it shows being one of the top performing method. And here I report the average area under the precision recall curve. Average across all the task we consider. And as validation procedure, we adopt a threefold cross-validation. Why we adopt area under the precision recall curve and not the standard AUC measure? Because for unbalanced problem, AUC, the area under the rock curve, is not informative as this measure. There are some works that recently have shown also this one. And so here the results are quite, our approach achieved the best results in all the three geo-ontology. Here BP is a biological process, one branch of gene ontology, molecular function the other branch, and cellular component the third branch. We run a separated experiment for each ontology by considering all the functions on that ontology. And so that's a preliminary experiment. We want to also try to understand if this multitask transformation is suitable also for feature-based prediction algorithm. We applied it on a label propagation that is a graph-based algorithm. But we want to try also if this kind of feature map is suitable, for example, when using support vector machines or when using any kind of regression. And also we want to investigate if this kind of transformation has any connection with the graph spectral theory to find other interesting properties that we may have in this kind of context, and also applying it to other problems in order to test if it works nicely also in other problems. That's all. Thank you.